fertilized ovule, because then only, like when the ovule gets fertilized, then only the seed is formed. Now, seeds are of various types depending upon whether they have consumed their endosperm or not. Okay. Now, the first type of seed is endospermic or albuminous seed. Now, here the endosperm is not completely consumed by the developing embryo, meaning the endosperm is persistent. It has not been consumed by the embryo. The examples are wheat, maize, barley, coconut, castor, and sunflower. So these are the endospermic or albuminous seeds. This is the name. Endospermic means that have the endosperm present. Then there is non-endospermic or ex-albuminous seeds. Here, the endosperm has been completely consumed by the developing embryo. So during the developmental process, the embryo completely consumes the endosperm, like it derives all the nourishment from it and grows. And that happens in the case of pea, bean, and groundnut. Then there is a very unique case of perispermic seed. In this case, nucellus is seen, like the persistent nucellus. Everyone remembers nucellus? The tissue that was surrounded by the integuments or protected by the integuments, that was the nucellus. Now, these seeds are those in which the nucellus remains are found. The residual persistent nucellus is called as the perisperm. Okay, whenever the nucellus is present, even after fertilization is done, even after every process has been completed, the nucellus, if it is present, we'll call it as perisperm. And in all those seeds which have the perisperm present in them, we'll call them as perispermic seeds. The examples for which are black pepper and beet. So these are the various types of seeds depending upon whether the endosperm has been consumed or not. Is this clear to everyone?
Yes, ma'am.
Yes, ma'am.
Okay, now moving forward, we are going to talk about the structures of the seed. Now the seed has a seed coat. Seed coat is the hard outer covering, and that forms a very protective layer for the seed. Seed coat is made by the hardening of the integuments of the ovule. The integuments that were present in the mature ovule, now what is their function? They become the seed coat. So see, slowly, you're going to see that the female reproductive part or the structures that were there in the embryo sac, all of them have their own functions to perform. And just like how we're talking about, like how nucellus is also there. Now we're talking about integuments. Now, whenever integument is there, now that integument gets hardened and forms the seed coat. This seed coat is tough and protective. Seed coat has two layers, outer testa and inner tegmen. Testa is the outer layer of the seed and tegmen is the inner layer. Now the micropyle, which is there in the ovule or the embryo sac, that remains as a small pore in the seed coat. You might have noticed if you have seen the seed of chana. Okay, if you see that what is it called? The gram. If you see it below, right below, like for example, if this is the seed, right below there is a pore-like structure, and that pore is actually the micropyle that still is present. Now, this pore helps in the entry of oxygen and water during seed germination, so that gaseous exchange and the availability of water is maintained with the help of this pore. Now, apart from the seed coat, the second part that is present in the seed is the endosperm. Now, if the endosperm is present, then that will be endospermic seed, just like how we discussed right now. It may be absent, then we'll call it as non-endospermic seed. Lastly, inside the endosperm, the embryo is there. Now, this embryo is responsible to give rise to the mature plant. Meaning, we know that whenever we want to grow a new plant, we use the seed. Now, when the seed is like put into the soil, we add water and everything. Then, through the micropyle area, oxygen goes there, water goes there, And because of that very reason, the embryo, the seed germinates, and the embryo gives rise to the mature plant. Now the embryo has the plumule, it has the radicle. Plumule gives rise to what?
Stem.
Yes, and radicle gives rise to?
Root.
Correct. Now we're going to talk about two very important phenomenon, that is dormancy and seed germination. Both of them are opposite to each other. Now dormancy, dormant means still. Okay, anything that does not move, does not perform any function. Now it is the state of inactivity of embryo when the seed is not able to germinate due to the absence of favorable condition. Meaning when, okay, if we have put the seed into the soil, now after that what will happen is that, okay, we have put it, but if we do not provide enough moisture, if we do not put it in a very airy place, then that seed will not germinate. And that process or that state wherein the seed does not germinate, it stays the same, that is known as dormancy. And okay, and during this time, in this time I've written, meaning during the time of dormancy, the moisture content of the seed decreases and reaches 10 to 15%. So there is a very gradual decrease of moisture content as well. Now talking about the next thing, that is seed germination. It is exact opposite of dormancy. It is the ability of the seed to give rise to the seedling in the presence of favorable conditions such as adequate moisture, oxygen, temperature, meaning whenever the appropriate conditions are present, then only the seed can germinate, or else it will stay dormant forever. Now, advantages of seed to angiosperm. Like seed is the result of sexual reproduction, and it is very important. Now, seed has better adaptive strategies for dispersal to new habitat. Whenever, like even if like we are eating any fruit and we throw away the seed in the garden, just like that, or the ground, later on, some way it will actually give rise to a new plant. So it has that ability to do so. It has sufficient food reserve for nourishing the young seedling because of the presence of endosperm. The embryo or the seedling, the young seedling gets nourishment. The seed coat which is present, it provides the protection to the young embryo because seed coat is a very hard, tough covering which surrounds the seed and it does not let the seed degrade very easily. It generates new genetic recombination. Why? Because first of all, it is a result of sexual reproduction. Anything that is a result of sexual reproduction will have variety of characters to them. So that is the reason why the seed will also generate new variation, like new types of plants. Seeds are very important for our agriculture as they show two very important properties. Seeds have two very important properties. One is dehydration, the second is dormancy. Now, dormancy is something we have already discussed about. Now, you might have noticed that if you ever go to the market, okay, to buy seeds for planting, they sell it in a packet, isn't it? They often sell it in a packet. How is that seed living? And those seeds are live. Like when you plant them, the germination happens. Seed. So this is a great property of the seed that we can dehydrate them. Their water content can be taken apart, and then also they will be, you can say live, or they'll be living. And as soon as you provide water to them, they will germinate. So just to increase their shelf life or storage life, dehydration is a very great property that they have. Like even after dehydration, even if you take out the water content that they have, they will live, they will not die. It's just that they will be dormant. They will not, you can say, express themselves very easily. Now coming on to the next, that is seed viability. It is the period for which the seeds retain their power of germination. Just like how we have studied about pollen viability, same way there is seed viability. Seed viability is the time period for which the seeds will retain the power of germination. Now we'll be seeing certain examples such as oxalis. Now the seeds of oxalis plant, they lose their viability within few months. So that is their viability period. Then there is lupine, the scientific name for which is Lupinus arcticus. It is the oldest seed which was excavated from the Arctic tundra. This seed germinated after 10,000 years of dormancy. So it was dormant for 10,000 years. Imagine, after 10,000 years, when water was provided to it, it germinated into the plant. Then there is another, that is the date palm. Phoenix dactylifera is the scientific name. This was excavated at King Herod's place near the Dead Sea, and this is viable for 2,000 years. So if we take the seed of the date palm, and even after 2,000 years, we want to plant it to get a new plant out of it or new germination. So then it will, even after 2,000 years. So the greatest dormancy period was of lupine, then comes the date palm. Up till now, any doubts, students?
No, ma'am.
Okay.
No, ma'am.
Now moving forward, see, the number of seeds in a fruit is equal to the number of ovules in an ovary. Because if you say ovules or if you say seed, it is the same thing. If you say fruit or if you say ovary, again the same thing. Always remember that the number of seed will never exceed the number of ovules, because ovules only turn into the seed. So if 5 ovules are there, 5 seeds will be present. If 10 ovules are there, 10 seeds will be present. It will not be more. Now, orchid's fruit contains thousands of tiny seeds. It means that it has thousands of ovules present initially. Now, there are certain parasitic species. They also contain many tiny seeds such as orobranche and striga. These are the examples of orchid, orobranche and striga. These are the examples for all those plants that have thousands of tiny seeds. Now we have discussed about seed. Let us discuss about the fruit. Now, just like how the ovules, they mature into the seed, same way ovary will mature into the fruit. The ripened ovary is a fruit. The wall of the ovary forms the wall of the fruit. The wall of the fruit is known as the pericarp. Okay, just like how testa and tegmen are the outer layers of the seed coat, right? These are the seed coat. The wall of the fruit will be called as the pericarp. Because see, if I talk about the mango, the peel that we remove, that is the pericarp. The flesh that we eat, that is the mesocarp. And then inside is the endocarp. Now, depending upon from where the fruit is developing, there are 3 types of fruit, 3 basic types. True fruit, false fruit, and parthenocarpic fruit. Talking about the true fruit, the fruit which develops from the ovary will be called as the true fruit, and the example is mango. Meaning, the ovary, when it gets ripened, it forms the fruit. Talking about the false fruit, the fruit which develops from other floral parts. Okay, some other floral parts and not from the ovary, those fruits will be false fruit. For example, apple, it develops from the thalamus. The bulgy portion of the flower, the thalamus, that gives rise to the fruit of apple. Okay, so it does not develop from the ovary, it develops from the thalamus. And also for strawberry, cashew nut, all of these develop from other floral parts and not from the ovary. Then talking about the third type of fruit, that is parthenocarpic fruit. The fruit which develop without the process of fertilization, meaning fruit is a product of fertilization only, but here fertilization is not happening, rather it is artificially developing. These are produced by the application of growth hormones like auxins. Everyone knows auxin? Yes.
Yes.
Very good. So, because of the application of these growth hormones, we can induce fruiting. And 100%, since they are not the product of fertilization, then they will not have seed also. So these fruits are seedless, such as banana. Banana is a parthenocarpic fruit. Clear? So these are the 3 different types of fruits depending upon where they develop from. Moving forward, the fruits can also be fleshy, such as mango, guava, oranges, and the fruits can be dry as well, mustard, groundnut. The only thing to remember over here that the fruits that we study in English, right? We study that certain things are fruits, certain things are vegetables. But remember, in terms of biology or in terms of sexual reproduction in flowering plants, the fruits are nothing but the product of fertilization. So whether it is mustard, groundnut, that beans is a vegetable, right? Peas is also a vegetable, but it will be considered as fruit. Okay. Now coming on to the last 2 topics. Finally, this chapter is going to end. The last topic is apomixis. See, I'm even pronouncing things wrong. Apomixis and polyembryony. Now, the production of seed, see, the production of fruit without fertilization was parthenocarpy. This process, the production of parthenocarpic fruit is parthenocarpy. This is the process through which you can make the seedless fruit. Now talking about apomixis. Now apomixis is the production of seed without fertilization. It is commonly seen in grasses and asteraceae. Like this apomixis is a natural phenomenon. In some plants, seeds are formed, but fertilization was not done naturally. And it is a form of asexual reproduction which mimics sexual reproduction. It is showing that, okay, somehow sexual reproduction is happening, but it is not actually sexual reproduction because sexual reproduction involves the fusion of gametes, which is not happening here. And it naturally occurs in some grasses and the members of the family Asteraceae. Now, the next thing is that the apomixis is of 2 types. Or you just talk about one type only. See, adventive apomixis. The embryo arises from the diploid sporophytic cell, such as nucellus or integument. Here, the embryo which is present inside the seed, that is being arise from the sporophytic cell, that is 2N, such as in the case of citrus, opuntia, mango. So apomixis can be seen naturally in some plants, but mostly seed is a result of sexual reproduction. Now talking about the next phenomena, that is polyembryony. Poly means more than one, right? More than 2. Embryony is referring to the embryo occurrence, that is more than one. So it is the phenomena of the occurrence of more than one embryo in a seed. Normally, one seed has one embryo, but when one seed has multiple embryo, we'll call that phenomena as polyembryony. Again, this is a phenomena that may occur on its own. Okay. Then comes apomictic polyembryony. By this method, many hybrid varieties can be made as they will have higher yield, vigor, and stress resistance. Meaning, we can actually induce polyembryony. Polyembryony is, we can get multiple seeds, multiple embryo, and also apomixis, seed without fertilization, because fertilization takes place with a lot of time. It requires time. So a quick solution is apomixis. Now, the production of hybrid is very costly for agriculture. So that is why apomixis is the solution to this problem. We can, like hybrids are made when 2 superior parents are crossed. But when that costs a lot of money. So apomix is something that naturally happens. Fertilization is not needed, meaning that much time is not needed. And the seed is also being formed. So it is very great for human use because it's not costly at all. Up till now, everything is clear to you people?
Can you explain adventive apomixis?
Adventive apomixis. See, normally, where does the embryo develops from? Embryo develops when the endosperm development takes place, and then
Yeah.
embryo forms it is present inside the seed, right? Ultimately, where is it coming from? It's coming from the ovule, isn't it?
Yeah.
But talking about the adventive apomixis, this embryo is not coming from ovule, rather it is coming from diploid cells. Okay, diploid sporophytic cells, forget sporophytic. Look at diploid cells. So diploid cells such as nucellus, integument. Integument is the outer covering. Nucellus is the nutritive tissue. Now the embryo is developing from them. Instead, it should have developed from the ovule. So that is adventive. Okay,
Yes.
clear?
Yes.
Do you people have any doubts?
No.
Are you people
Is
happy
the
that
chapter over?
No.
the chapter is over? Why haven't you switched on your camera today?
Because the focus mode is not on.
Focus mode is on. It's not.
Like,
Wait.
but I can see.
Now can you see people?
No.
Switch it on. Oh, I did not realize that. Okay. All right. So, where did you people left off in the previous class? From where do you have to write down?
Embryo development, ma'am. We only wrote 3 lines of it here.
Embryo development. I'm so happy that this chapter is
Yes, ma'am. 1 chapter completed.
one chapter completed. Yep. This part, right?
Yes, ma'am.
Okay. You people can switch off your videos, write comfortably.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Done, ma'am.
Done, ma'am. Done, ma'am.
Everyone's done?
Yes, ma'am. Done, ma'am.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Yes, ma'am.
Done, ma'am. Can you scroll towards the right? Done, ma'am.
Everyone's done?
Yes, ma'am.
Done, ma'am.
Done, ma'am.
Done, ma'am.
Done, ma'am.
Done.
Done, ma'am. Done, ma'am.
1 minute, ma'am. Done, ma'am. Done, ma'am.
Done, ma'am. Done. Done, ma'am. Ma'am,
Yes.
what is the meaning of excavated? Excavated.
Excavated. See, excavation is when people do the digging while they're on, what do you call it as? Like archaeological stuff, the digging that people do. So as to find
Oh,
stuff that are, buried under the ground for thousands and thousands of years. Excavation,
oh.
so when people dug it and found this out.
Okay, ma'am. Thank you. Done, ma'am.
Done, ma'am.
Done, ma'am. Ma'am,
Yes.
pericarp is also called the epicarp, right?
Correct.
Done, ma'am. Ma'am, can you move to the right side?
Done, ma'am.
Done, ma'am.
Done. Done, ma'am. Done. Done, ma'am.
Done, ma'am. Done, ma'am.
Done.
All right, everyone's done?
Yes, ma'am.
Yes, ma'am.
Okay, good. Now, with very deep and sad feelings in my heart, I would like to inform you people one thing that we had missed in this chapter. That is artificial hybridization. I did not teach you that. Okay, that part is there in this chapter, and I did not teach you that. So let me give you a quick brief. See, it is very easy. Main thing, what happens, as you remember, that it will be much more ideal for us to avoid self-pollination in plants and induce cross-pollination in plants, right? Now talking about artificial hybridization. Wait, let me just open the NCERT PDF. So till that is opening, see, basically what is the case is that when there is a bisexual flower, when there is a flower which has both male and female parts in it, then what we do is to avoid the male part from fertilizing the female part, we cut the anthers. Imagine a bisexual flower is there. A bisexual flower has stamen and pistil. Now, from the stamen part, we are cutting the anther. We are removing the anther so that that anther cannot fertilize this pistil of the same flower. You getting my point? So this process, what we do, that is actually the process which is involved in artificial hybridization. Wait. Okay, also,
The anther won't grow back.
no, it won't. It won't. It's not a regenerative thing. It is an organ. If you cut an organ, the organ does not grow back, right? So the answer will not grow back. And with that being said, most of the times what happens is that even after the anther is being snipped off, then also it is actually helpful in the process that people cover it, that to even be like 100% sure that the contamination does not happen. Because if the pollen comes, if the same pollen comes and lands on the stigma, then 100% the fertilization will take. So just wait, let me share that with you. What is shown on the screen of your people? What can you see on your screen?
My notebook, class 2,
Oh, my
all
notebook.
the topics.
Okay. See this. This is your NCERT books chapter. Have you people downloaded your PDFs to at least get the idea as to what is there or no?
I read the textbook once, but we didn't complete a chapter that time. I did half.
So we have discussed every single thing. I just want to see where that part is, because even I am forgetting that. I have not included it in my notes.
So your last batch, you didn't teach them?
See, such a beautiful flash. What, the last part?
The last batch, you didn't teach them that?
I did. I used the PDF only. This, the notes that I am sharing with you, it is the one that I have initially formed, like made these notes. Okay, so is my screen visible to you people?
Yeah, it's blurry.
It's blurred.
Why is it like this? I don't understand.
Is it long, the topic?
It's not long. That is why I am, I'm not starting a new chapter. I would rather finish this chapter. See, nothing is even loading. Oh, there it is. No. Even that's not loaded in my phone also. Just one second. Give me 1 second.
Sure.
Okay, see, if this is not loading, it will now. Outbreeding devices, and then there is artificial hybridization. Yes, yes. If it loads, see, artificial hybridization, it is a technique wherein we want to do the pollination by us. Okay, like we want to aid the process of pollination so as to get what we desire in a plant. Maybe, for example, if there is a plant A, okay, and then there is a plant B. Now, plant A has certain characters such as it is very resistant to heavy rainfall. It will not get destroyed when heavy rainfall occurs. Then plant B is resistant to insects. Okay, meaning if even if insects come and infect this plant, then what will happen? This plant will not get affected by it. So now we want the better qualities from both of these, and we want to combine them in order to form a new plant, and that new plant will have the good qualities of both of these things. Okay, so this is something that is 100% a man-made experience, a man-made experiment, and we do it. It is like very famous, and this is known as artificial hybridization. Now it has certain mechanisms. See, artificial hybridization is one of the major approaches of crop improvement program. In such crossing experiments, it is important to make sure that only the desired pollens are used for the pollination. Now, if this is the female plant, okay, now we want the pollen from this A, not C, right? Now, this C can potentially pollinate this plant. Now, we want to prevent that so that we can only pollinate the B from A, right? So, with this, we know that we need to use the desired pollen grains, and the stigma should be protected from contamination from unwanted pollen, because even by mistake, we do not want the wrong type of pollination to happen. Now, this is achieved by emasculation and bagging techniques. These are the two techniques that are used in artificial hybridization. If the female parent bears bisexual flowers, a flower which has male and female both, removal of anther from the flower bud before the anther dehisces using a pair of forceps is necessary. Because once the anther matures, it will burst, right? So when it is in a bud situation, when the flower is closed, that is a bud situation, right? When the flowers are closed. So before the blooming of the flower, what we do, we take forceps and with the head. Since it is a flower, so it is very easy to disintegrate it, right? To break it. So we take forceps, and with the help of that, we pinch off the anther. This step is emasculation, cutting the head off. Okay. Emasculated flowers have to be covered with a bag of suitable size, generally made up of butter paper, to prevent contamination of its stigma with unwanted pollen. So after the anther has been cut, we put a bag around it and we tie it so that even if there are some left pollen, it will fall into the bag and not onto the stigma. This process is known as bagging. When the stigma of bagged flower attains receptivity, meaning stigma becomes receptive, it becomes functional, or you can say mature. Now the mature pollen grains, which is collected from the anther of the male parent, whatever desired plant is there. Now when we collect, like how do we collect? We, for example, we take any substance, or we take a brush, like how paint brushes are there. We take a brush and we lightly try to dust it, like try to dust off the pollen onto the brush. Now the brush has the pollen. Now we take that brush and put it on the stigma. Okay, so those are dusted on the stigma, and then the flowers are rebagged and the fruits are allowed to develop. Meaning we are doing the process of pollination. So this is artificial hybridization. Artificial, why? Because we are involved. It's not a naturally occurring process. Now, if the female parent produces unisexual flower, there is no need for emasculation. The flower is only like the female part is present or the male part is present, then we don't need to emasculate. The female flower buds are bagged before the flowers open. Now, in these cases, still we want to prevent the contamination with unwanted pollen, so we are again bagging it. When the stigma becomes receptive, pollination is carried out using the desired pollen, and the flower is rebagged so that no more of the foreign pollen can come. So, for artificial hybridization, it is a very important method which is used in crop improvement. Why do I say crop improvement? Because it involves the superior qualities from parents to come into the offspring. So now this crop which we will be getting will be much more superior than both of the parents, because it will have the quality of both of them, right? So this is why artificial hybridization has taken agriculture to whole new levels. People like study this thing. Okay, so this is your whole entire artificial. Now we have completed chapter. This part you've understood? Sure?
Yes, ma'am.
Okay, very good. So the next chapter that we will be starting will be human reproduction. Now this will, I hope so, not be as boring as this chapter. And again, I apologize in advance because this chapter is also very long. It is humongous. It is bigger than sexual reproduction, I think so. But it is good. It's like not boring. Okay, and is my screen sharing for you people?
Yeah.
What can you see on your screen? Human reproduction?
No. The
No,
artificial.
artificial hybridization, ma'am.
Oh. It has been a while since I've been using this. Still, I can't navigate around. Okay. Anyway, I'm not going to start the new chapter today. You people tell me if you have any doubts or anything. Do you want me to send you the PDF so that you people can read it?
NCERT thing. Okay, yeah.
Yeah? Or you can download it also. It's like available on internet.
Okay.
Okay, and this chapter is completed. So now I want you people to be done and dusted with this chapter. Read your NCERT, whether you download the PDF or I'll send it. Don't worry, I'll send it also. You read the PDF, you read my notes. Everything that's there in the PDF is there in the notes, vice versa. This is the first chapter. We've uh, put on a good knuckle into the whole entire syllabus. Now I want you people to prepare right like from now. We know that boards is like a long way far from us, but still we'll be so prepared that during the time of boards, the revision will actually be a revision and not the studying portion, right? So this chapter is done. The questions, are you people getting the questions after the class? Are you people able to solve?
Yeah, most of them are from notebook only, ma'am.
Okay, good. Okay.
So all of them are from notebook, ma'am.
Okay, all of them are from notebook. Yeah,
You
it's good. It's
just,
very NCERT based only. It's like nothing from the outside is there. Okay.
you just read today's notes, you can solve. You can just read today's notes, you can solve those questions.
Okay, that's perfect. So now you'll get the rest of the questions for today's homework, and this chapter is done. I'm so happy. It took a really long. You know why it took such a long time? Is because initially we had just 1 hour classes.
Yeah.
If we had 1.5
And also
hours,
because of writing notes.
yeah. So that is why, like, see, now we have like 1.5 hour classes and it is very sufficient. Like how we used to have in class 11, isn't it?
We had 2 hours too.
Yeah, we had even 2 hours. Oh God. Yeah.
I
Okay.
missed 11.
So we have, how many times am I going to say the same thing that we have completed the chapter? See how happy I am. So now it's your duty to learn everything that is there in the chapter. And as far as your answering